Hello, I'm Reza and you are watching Hubert short videos. In this short video, we are going to talk about the endomembrane system, the two member of endomembrane system in eukaryote cells, the Golgi apparatus and endoplasmic reticulum. Let's start with endoplasmic reticulum. The endoplasmic reticulum is an organelle in the cell that puts up from the nucleus. So let's say it's the nucleus of the cell. It has a nuclear envelope, which is double membrane, and this quite large structure connected to the nucleus is called endoplasmic reticulum. Notice that the space in the nuclear envelope is continuous with the lumen or space of the endoplasmic reticulum. So what does the endoplasmic reticulum do? Well, endoplasmic reticulum actually has two parts to it. It has one part that known as a rough endoplasmic reticulum. You can see here in the picture it is colored in blue, and that has a bunch of ribosomes. Those are blue dots that you see on the surface of rough endoplasmic reticulum, or RER. Here is the ribosomes. And then endoplasmic reticulum has a smooth part that does not have any ribosomes on it. And you can see here in this picture the smooth endoplasmic reticulum colored in kind of orange color. Let's start with a smooth endoplasmic reticulum. What does it do? A smooth endoplasmic reticulum synthesizes lipids, including those that will end up being part of the cell membrane and those that are secreted from the cell, for example, steroid hormones. A smooth endoplasmic reticulum also metabolizes carbohydrates, and it also aids in the detoxification of drugs and other toxins. So let's go to the rough endoplasmic reticulum. As we mentioned, it has ribosome, which means it's the site of protein synthesis. But we know that there are also ribosomes that are in the cytoplasm. So what's the difference between those proteins that are translated in the cytoplasm and those that are translated in rough endoplasmic reticulum? So let's split protein synthesis into two parts. We have those that are made in the cytoplasm and those that are synthesized in rough endoplasmic reticulum. They might end up in mitochondria. They might end up in peroxisomes, or they just might stay in cytoplasm. Maybe that's where they belong. In contrast, proteins that are synthesized in the rough endoplasmic reticulum will either secreted into the extracellular environment, or they will end up becoming into raw proteins in the cell membrane. Or they might remain in the endoplasmic reticulum, Golgi apparatus, or lysosome. And you'll see in a few moments why I'm grouping those three organelles together. And I just want to mention as a side point that another thing that happens in the rough endoplasmic reticulum is post-translational modification of proteins. For example, the formation of disulfide bridge in proteins that happens in the rough endoplasmic reticulum. Okay, let's go back to the protein synthesis that happens in the rough endoplasmic reticulum. So, proteins that are secreted from the cells or that become part of the cell membrane follow what we call the secretory pathways. The secretory pathway describes the pathway of a protein takes from when it's synthesized until it leaves the cell or become the part of cell membrane. But you might be thinking, how does a protein know that it's supposed to be following the secretory pathway, and therefore that it should be synthesized in rough endoplasmic reticulum, as opposed to the cytoplasm? So the answer to that question is that all proteins begin to be translated in cytoplasm. But those that need to follow the secretory pathways have what called a signal sequence. That signal sequence is detected early on translation and will cause the polypeptide that being synthesized to be pushed into the rough endoplasmic reticulum where translation is complete. In order for us to understand the secretory pathway, we need to talk about another organelle. That organelle is called Golgi apparatus. The Golgi apparatus is an organelle that found near the endoplasmic reticulum, and it's basically is a group of sacs that are stacked together. Each section of these sacs has specific names and specific functions. We have a cis face, which is directed toward endoplasmic reticulum, and vesicles carrying protein cargo from endoplasmic reticulums enter the Golgi apparatus from the cis face. In the middle of the Golgi apparatus, we have middle face, which has a specific function that we are going to mention to it in another videos, in upcoming videos. And we have trans face, which the vesicle export from Golgi apparatus occurs here. So the exported vesicles from Golgi apparatus, from cis face of Golgi apparatus, will direct it towards membrane or lysosomes. So what happens in Golgi apparatus? Number one, the Golgi apparatus modifies proteins that are made in rough endoplasmic reticulums. Number two, the Golgi apparatus sorts and sends proteins to their proper destinations. And number three, the Golgi apparatus synthesizes certain molecules that are needed to be secreted from the cell. Thank you for watching this video. We will discuss the secretory pathway in detail in upcoming videos. See you in Hubert.